Welcome back to the SANS Channel podcast, where we discuss the coolest scientific papers and preprints of our day.
Great to be back.
And hey, if you're digging these deep dives into the latest research, please give that like button a tap and subscribe. It really helps us keep bringing you more of this stuff.
Yeah, we really appreciate the support. It lets us share all these, fascinating discoveries.
So today we're plunging into a paper from MBO Molecular Medicine. It's titled, A Coordinated Multi-Organ Metabolic Response Contributes to Human Mitochondrial Myopathy.
Right. So mitochondria, as many listeners probably know, are the tiny powerhouses in our cells.
They generate energy. And mitochondrial diseases, well, they involve problems with that energy production.
Exactly. And tissues that are really energy hungry, like our muscles, they often bear the brunt of this.
Which leads to mitochondrial myopathy, basically, impaired muscle function.
And we know a fair bit about the genetic quirks, the mutations that cause these muscle issues. We found many of the genes involved.
Right. The genetic basis is becoming clearer. But what's been sort of fuzzier is why those specific genetic problems lead to the metabolic breakdowns that actually cause the muscle to degenerate. The biochemical how.
That's the key question this paper tackles. They took a really interesting approach, looking at metabolic changes in two groups.
Okay, who are they studying?
First, human patients with a specific type, called MERF myoclonic epilepsy, with ragged red fibers. And second, a mouse model.
Ah, the mouse model. Always useful. What was special about this one?
It had a muscle-specific mitochondrial defect. They knocked out a gene called COX10 just in the muscle tissue.
So you can isolate the effects originating from the muscle's mitochondrial problem?
Yeah,
clever.
Exactly. And what they found were some, pretty surprising similarities between the MERF patients and these mice, and potentially some clues for treatment down the line.
Okay, let's get into it. What was one of the first big findings? Something that maybe made them scratch their heads?
Well, right away, in the muscle tissue itself, they saw something quite striking. In both the patients and these COX10 knockout mice, the muscle showed what they called a starvation-like metabolic shift.
Hang on, a starvation response in the muscle, but these patients and mice, presumably they're eating normally, right?
That's the apparent paradox. It sounds weird, but if you think about it from the cell's point of view, if the mitochondria, the powerhouses, aren't generating energy properly, the cell might be sensing an internal energy crisis, regardless of external food supply.
So it triggers these emergency survival pathways, the kind usually activated during actual starvation.
That seems to be the idea. And specifically, this involved the muscle cells ramping up the breakdown and oxidation, the burning of amino acids for fuel.
So using the protein building blocks as emergency energy, like burning the furniture to stay warm?
Kind of, yeah. It's trying to find any available fuel source when the main ones aren't working well.
Which amino acids were involved? Did they pinpoint specifics?
They did. They found higher levels of several amino acids in the muscle of the MERF patients. Things like the branched-chain amino acids, BCAAs.
Right.
You hear about them in sports supplements.
Right.
And also glutamate, asparagine, and tryptophan were elevated.
Interesting mix. And the mice, did they show the same thing?
Pretty much, yeah. Early on in the disease progression, in those COX10 knockout mice, they saw increased activity of the enzymes needed to break down glutamate, ALT2, GOT2, OGDH. These key enzymes were upregulated.
Okay, so the muscle is definitely kicking its amino acid metabolism into high gear. Was this actually helping, though? I mean, even short term?
It looks like it was, initially at least. They did a neat experiment where they pharmacologically inhibited glutamate breakdown in the mice.
And what happened?
Their exercise endurance got worse.
Okay, so messing with that pathway actually harmed their function.
Exactly. It strongly suggests that this push to oxidize amino acids, especially glutamate, is a really important compensatory mechanism. It's the muscle trying its best to make up for the energy deficit from the faulty mitochondria.
That's a crucial point. It's adaptive, not just a random side effect. But, like you hinted, really not a perfect long-term solution. The paper talks about downsides, right?
Absolutely.
Yeah.
This initial muscle response, this starvation-like state, seems to be the first domino. It sets off a whole cascade of metabolic disturbances that ripple out across the body.
So it becomes a multi-organ issue, not just contained within the muscle.
Precisely. The problem spreads, metabolically speaking.
What other organs started getting involved? What was the downstream effect?
Well, the liver's behavior changed quite dramatically. It started pumping out more glucose via gluconeogenesis.
Making new sugar, basically, to try and feed the body.
Yes. likely trying to compensate for the perceived energy crisis, and probably using some of those amino acids coming from the muscle as building blocks for the glucose. The liver also ramped up urea production.
To handle the extra nitrogen from breaking down all those amino acids?
Exactly. You get ammonia when you break down amino acids, and the liver converts that to less toxic urea for excretion. And on top of that, the liver started producing more ketone bodies.
Another alternative fuel source. So the liver is really trying to manage this situation. You mentioned hormonal signals were involved too.
Yes, this whole coordinated multi-organ response seems to involve signaling molecules like leptin and, significantly, glucocorticoids.
Leptin, the satiety hormone, and glucocorticoids. Those are stress hormones like cortisol.
Correct. They seem to be key players orchestrating this broader metabolic shift.
And could you see signs of this systemic change in the blood work of the human MRF patients?
Oh, yes, definitely. The patients had elevated levels of lactate, alanine, another amino acid involved in glucose transport, urea, as we mentioned, and also something called guanidinosuccinic acid.
All pointing towards this bigger body-wide metabolic upheaval.
It really underscores that it's not just a muscle problem in isolation.
Okay, so we've got this cascade. Muscle senses starvation, breaks down amino acids, signals the liver changes its output, hormones get involved. How does all this ultimately connect to muscle wasting? That's such a key feature of these myopathies.
Right, that's the critical link. How does this metabolic remodeling cause the muscle loss? The study dug into that. In the mice, they saw that the activity of mTORC1 was reduced.
mTORC1, that's like the master switch for building proteins, right?
Yeah.
Protein synthesis.
Exactly. It's a key regulator. And its activity dropped right around the time the mice started losing weight.
So less building of muscle protein. That would definitely contribute to wasting.
For sure. If you're not building new proteins, the muscle mass will decline.
Okay.
But it wasn't just reduced building.
What else? Increased breakdown.
Yes. Later in the disease progression in the mice, they saw signs of increased autophagy.
The cellular recycling system.
Right. Normally it's good, cleans up damaged stuff. But if it goes into overdrive, it can start breaking down functional muscle proteins too. They also found higher expression of genes that specifically inhibit mTOAC1 like Cespin2 and DDIT4.
So it's a double whammy, putting the brakes on building muscle and potentially accelerating its breakdown.
It paints a clearer picture of the mechanisms behind the wasting, yes.
Now, the study also mentions the ISRMT, the mitochondrial integrated stress response. What's that about?
The ISRMT is basically a cellular defense program. It gets activated when mitochondria are stressed or damaged. It's the cell trying to cope and ideally fix the problem.
Like an internal alarm system for mitochondrial trouble.
Pretty much. And they found clear evidence that this ISRMT pathway was switched on in the muscle of both the mouse model and the human Mirage patients.
Okay, so the cells definitely know something's wrong with their power supply. What does activating ISRMT actually do? What changes?
Well, they saw increased levels of upregulation of key ISRMT proteins. Names like ATF4, MTHFD2, ASNS, HSP60. These guys orchestrate the response.
And does this response send signals outside the cell too?
Yes, that's crucial. The ISRMT activation led to increased production and release of signaling molecules, notably FGF21 and GDF15.
Ah, FGF21. We hear that name a lot with mitochondrial disease. It's often used as a biomarker, right?
Exactly. It's a well-known indicator, and this study confirmed it was elevated in both their mice and the human patients. It's like a flare signal sent out by the stressed muscle.
What else does the ISRMT do? Anything potentially protective?
Interestingly, yes. It also seemed to activate pathways for making antioxidants, specifically glutathione or GSH.
So trying to combat the oxidative stress that often comes with mitochondrial problems.
That seems likely, yeah. It's a multi-pronged response trying to manage internal damage like oxidative stress while also signaling outwards with molecules like FGF21.
Okay. Now, what about fat metabolism? You mentioned ketone bodies earlier, but the paper also highlighted issues with how fat itself was being handled.
Right. This is another complex piece. It looked like the body was trying to use more fat for energy. They saw increased fatty acids in the blood and muscle.
Which makes sense if other fuel sources are problematic.
It does. But the process seemed inefficient, like it was hitting a bottleneck. They found a buildup of specific fat intermediates inside the muscle cells, things called acylcarnitines and 3-hydroxyacylcarnitines.
A buildup usually means something's not flowing properly downstream. What's blocking the breakdown?
The accumulation of these specific molecules points towards a problem with fatty acid beta oxidation, the process that breaks down fats within the mitochondria. The researchers suggested this might be due to a shortage of a crucial cofactor called NAD+ inside the mitochondria.
NAD+ is vital for so many metabolic reactions, including fat burning.
Exactly. If you don't have enough NAD+ in the right place, you can't efficiently burn the fat. And consistent with this, they actually saw lipid droplets, little blobs of fat accumulating in the muscle tissue.
So the fuel is there, maybe even piling up, but the engine can't burn it properly because it's missing a key component like NAD+ delay.
That's a good analogy, yeah. Struggling to use both glucose and fats efficiently.
Okay, one more connection to unpack. The link between fat loss, leptin, and those glucocorticoids, stress hormones. How did that play out?
This was really intriguing. In the mouse model, as the disease progressed, the mice lost body fat. And as their fat stores dwindled, their blood levels of leptin also dropped.
Lower fat mass, lower leptin. That makes sense.
Right, but the consequence is that low leptin can act as a signal to the brain in the adrenal system to increase the release of glucocorticoids.
The stress hormones. So fat loss triggers more stress hormone production.
It seems so in this context. Indeed, both the mice with increased corticosterone and the human mirth patients with increased cortisol had higher levels of these glucocorticoids.
Were these hormones just bystanders, or were they actually contributing to the problem?
They seem to be contributing. The really compelling part was an experiment where they blocked glucocorticoid signaling in the mice.
What happened when they blocked the stress hormones?
They saw some significant benefits. There was some improvement in muscle strength, and crucially, the mice lost less body fat.
Wow. So the stress hormones themselves appear to be driving some aspects of the muscle maintenance and the metabolic dysregulation, including the fat loss.
It really suggests they are part of the pathogenic process, not just a consequence. It's this feedback loop involving fat, leptin, and stress hormones that exacerbates the condition.
Man, it really is this complex, interconnected web, isn't it? Starts with the mitochondria and the muscle, but then pulls in the liver, fat tissue, hormonal systems. A truly coordinated multi-organ response, just like the title says.
Absolutely. It drives home that you can't just look at the muscle in isolation. The muscle's initial attempt to cope sends out ripples, signals that profoundly affect how other organs function, and that interplay defines the overall disease state.
Okay, so let's try and boil this down for everyone listening. The main takeaway seems to be that these mitochondrial muscle diseases trigger this big, coordinated metabolic shift across many organs.
Right. It starts with that sort of starvation-like response in the muscle breaking down amino acids for fuel.
But that initial fix leads to wider problems, imbalances in the liver, issues with fat burning, muscle wasting, fat loss.
And things like the cell's own stress response, the ISRMT, and those hormonal changes involving leptin and glucocorticoids are deeply involved in driving this whole cascade.
And importantly, understanding these steps, these metabolic pathways, might give us new ideas for treatments, right? Maybe targeting amino acid use, or maybe modulating that glucocorticoid response.
Precisely. It opens up potential therapeutic avenues beyond just trying to fix the underlying mitochondrial gene defect, which is obviously very difficult. We might be able to intervene at the level of these metabolic consequences.
Yeah, it really makes you wonder, could tweaking these early metabolic shifts be a way to prevent the worst outcomes of these diseases?
It's a hopeful thought, and it raises that broader question you mentioned.
Right, like what other conditions, maybe things we don't normally associate with mitochondrial disease, might actually share similar kinds of underlying metabolic crosstalk between organs?
Yeah. It highlights just how interconnected everything is metabolically. A problem in one place really can echo throughout the entire system. Fascinating stuff.
Absolutely fascinating. Well, that was a really thorough deep dive. We hope you all found that as interesting as we did.
Hope so.
And one last time, please don't forget to like the video and subscribe if you want more content like this. We'll see you next time.